don't forget that you need a broth of your unknown for tomorrow. The reason we want a broth is because when we inoculate those thioglycolate tubes, what we want to do is we want to just put a little bit of growth right at the very top of the tube. We can't really kind of knock it around like we normally do to get growth off of a slant from a root, because you're going to kind of aerate the media. So it's easier to inoculate those with the broth culture. And then a broth is fine for doing your plates for the gas bag and for the candle jar and for all these salt plates and things like that. So don't forget to do that. Next up we'll probably get started on that today actually a little bit, is material called control of growth, and that should be in Dropbox as well, with PowerPoints and the lecture notes. Any questions? Okay, I started the nitrogen cycle and then realized that I kind of neglected to say something in more detail about the chemoautotropes, and so I ended up backtracking a little bit. So let's go ahead and finish up talking about the chemoautotropes, and then we'll get back into the nitrogen cycle, and I'll do just a brief review of what we covered previously, and then get into the meat of the nitrogen cycle. So remember that with the microbial world, there are several different strategies of metabolism you can use. And in the microbial world, we typically characterize microorganisms based upon their source of energy and their source of carbon. Okay? And so in the eukaryotic world, we really just have these two patterns pretty much, photoautotrophy and chemoheterotrophy. But in the microbial world, we've got another couple of different combinations, and the one that we're interested in right now are the chemoautotropes, or the lithotropes. So these are the organisms that use entirely inorganic sources of both carbon and energy to sustain themselves. Because remember, metabolism's got to satisfy two needs. It's got to provide carbon skeletons, and it's got to provide energy in the form of ATP. And chemoautotropes get all those from inorganic compounds. One of the chemoautotropes that's particularly of interest in a number of ecological microbiological studies are the methanogens, and these particular bacteria are archaea bacteria. Now that doesn't mean that all of the chemoautotropes are archaic. This particular example that I'm going to give you are archaea bacteria. But what methanogens are able to do is they are able to extract energy in the form of electrons from hydrogen gas. And so they use an electron transport chain, for example, to strip off electrons from hydrogen gas. And they use that to power an electron transport chain, which will then produce ATP by chemiosmosis. But then additionally, they are able to use some of those electrons in the process of carbon fixation. Remember, if you're an autotroph, if you're using inorganic CO2 as your source of carbon, you've got to fix that into an organic form. What's the name of the cycle whereby carbon fixation takes place in the eukaryotic world, plants? Remember? The light-dependent reactions produces basically the ATP and the electrons, and then CO2 is fixed into carbon, or carbohydrate, in what cycle? Remember? That's the Calvin cycle. Okay. Well, some autotrophic organisms have a sequence of events that's very similar to the Calvin cycle. Some have other mechanisms of CO2 fixation. And these chemoautotropes have some different mechanisms. But the bottom line is that they've got to be able to reduce CO2 into carbohydrate. And the source of those electrons is this inorganic hydrogen. So they're really milking the hydrogen for electrons to both reduce CO2, in this case the methane, and then also using electrons stripped off of the hydrogen to generate an electron transport chain hydrogen ion gradient, and using that to make ATP. So they're pretty versatile organisms. You can find these methanogens in a lot of environments. Typically, these chemoautotrophs live in anaerobic environments. So you can find huge communities of buried under layers of mud. We're just finding out now that under the ocean seafloor, there are incredibly diverse communities of these chemoautotrophs living underneath the sea, underneath the seafloor. In fact, some people estimate that if you count a organism as a cell, in the case of unicellular organisms, that about a third of all living organisms live buried underneath the seafloor. There are these incredibly diverse communities of these bacteria, particularly the archaean bacteria that are chemoautotrophs living underneath the sea. And that has some ecological significance because these methanogens can produce methane as a result of their biochemical processes. And that methane can become compressed under the sediments and form these ice-like crystals of solid methane called methane hydrate. And it's kind of interesting, when they dig these things up, it looks like a big frozen snowball. But you can light a match to it and it'll start burning because it's actually a compressed methane gas. Well, there's some real concern about this because as ocean global temperatures increase due to global climate change, there's fear that these methane hydrates that are trapped on the bottom of the ocean are going to start warming up, and the solid methane is going to be released into gaseous methane. And as that bubbles up through the ocean and gets into the atmosphere, methane is a really potent greenhouse gas, and that's going to continue to contribute to global climate change. And so that can lead you into one of these really nasty positive feedback loops where the problem basically makes itself worse. And so as we get more warming, more of these methane hydrates become gaseous. That causes more warming, more become gaseous. So this is a considerable problem that the world could be facing. And it's all as a result of the metabolism of these little single-celled chemolithotrophic organisms. You can find these organisms that are chemolithotrophs in hot springs. You can find them in swamps. I don't know if you've ever seen in a swamp where gas will come bubbling up to the surface of the swamp. It's called swamp gas. And that's methane produced as a result of this particular type of metabolism. We can find these type of organisms in intestines of both humans and cattle. The rumen of a cattle, where the cellulose digestion takes place, contains a number of these types of organisms, and cows continually are belching methane. We have some methanogens in our gut, and we produce methane as a result of their metabolism as well. But for our interest, one of the places we also find these particular organisms, the chemoautotrophs, are in the soil. And they do play an important role in the nitrogen cycle. So let's get back to the nitrogen cycle. Remember from this particular slide up here that the nitrogen cycle consists of 4 basic steps: nitrogen fixation, ammonification, nitrification, denitrification. And all of these steps are carried out by the metabolism of different groups of microorganisms. We already talked about nitrogen fixation and symbiotic and non-symbiotic. And nitrogen fixation is where the N2 gas, which is notoriously unreactive, it's a very stable molecule, is turned into ammonium, which can be a plant nutrient. Likewise, ammonification can produce ammonia as living or dead, more likely, organic material is decomposed. And during the decomposition process, the amino acids are deaminated, and that ammonia that's released can be released into the soil. But plants have to have ammonium to grow. Okay? That's the form of nitrogen that they need to be able to then make amino acids, make nucleic acids, and they've got to make those things in addition to carbohydrates if we're going to make new cells and grow. So the other 2 important parts of this nitrogen cycle are nitrification and denitrification. So let's take a look at those for a second. Okay, in nitrification, what happens is the ammonia that is produced either by ammonification or by nitrogen fixation can be converted back into different compounds. Now, remember where that ammonia, where that nitrogen and ammonia comes from ultimately. It comes from the atmosphere. And if we're going to have a self-sustaining cycle, we need to get that nitrogen back into the N2 gas form eventually, right, to keep the system going in a cycle. Okay? So this is one of the steps that helps to return the nitrogen ultimately back into nitrogen gas. Nitrification converts the ammonium to either nitrate or nitrite, depending upon the species of bacteria you're talking about. And these particular organisms are these guys. These are the lithotrophs. So what are they doing? They're oxidizing that ammonia. They're ripping electrons off of it. And in the process, the ammonia is converted into other nitrogen forms that are more oxidized, like nitrate or nitrite. And then they're also using some of those electrons to fix carbon so they can make carbohydrate. So without these lithotrophs, the nitrogen cycle would stop. It would become short-circuited. And we wouldn't complete that conversion of nitrogen back into nitrogen gas again. Incredibly important. This is a very important step in sewage treatment. In sewage treatment, we use natural microbial activity to convert waste matter back into inorganic substances in the process of water purification. And this is a naturally occurring process, okay? When you dump fecal matter into a body of water, there are naturally occurring organisms in there that will break that organic material down into inorganic substances and purify the water. Now the problem becomes is when we start discharging too much waste into water and we overwhelm the system. And that's when we have to supplement it with artificially constructed sewage treatment ponds, for example. But microbial decomposition is still a very important part of both natural and human engineered sewage treatment processes. And one of the metabolic processes that's really important is this nitrification process. Because ammonia is toxic. We've got to remove the ammonia from the water. It's going to be safe to drink. But then that ammonia is converted into nitrate and nitrite, and those are a little bit toxic as well, not quite as bad as ammonia. So different groups of bacteria then can convert the nitrate and nitrite back to nitrogen gas again. Now that's also essential in the nitrogen cycle. So in some respects, the sewage treatment process is kind of the nitrogen cycle in an aquatic environment. Okay, denitrification, what does that do? That takes these compounds, these products of chemoautotrophy, and converts them finally back into atmospheric nitrogen. How do we do that? That happens during anaerobic respiration. So in anaerobic respiration, we have an electron transport chain. That electron transport chain is removing electrons from reduced coenzymes like NAD and FAD. And we're generating an hydrogen ion gradient during this electron transport chain, which is making our ATP. But then the electron's got to be dumped somewhere. And remember, in anaerobic respiration, it's not oxygen. There's no oxygen present. And instead, we use some oxygen-containing molecule like nitrate. And maybe we will reduce it, if we're one organism, into nitrite. There are other bacteria that can then take that nitrite that's produced, and further converted into other, more reduced compounds. So as a result of anaerobic respiration and the metabolism of several different species, nitrate can be reduced to nitrite, nitric oxide, nitrous oxide, nitrogen gas. There are all kinds of different possible outcomes. Typically what happens is one organism does nitrate to nitrite, another organism then takes that waste product, basically nitrite, and uses it down here and makes nitric oxide, and then the next organism uses nitric oxide and makes nitrous oxide, and then another organism will take that and convert it into nitrogen gas. And there is another little other pathway that can occur, which can result in nitrate being reduced directly into ammonia. Now, as far as the nitrogen cycle is concerned, this is kind of the final process that gets our nitrogen back into the atmospheric form again to complete the cycle. Now, we're going to do this in lab. It's going to be a few weeks. And we're going to do a test which has a series of steps called a nitrate test. And what we want to see is, can an organism, through anaerobic respiration, reduce nitrate into something? The two things we really will be looking for is, can your organism use nitrate as an electron acceptor and produce nitrite? And that's very diagnostic. Another thing we will look at is to see if your organism can take nitrate and reduce it into ammonia, because that's pretty diagnostic. These other compounds right here are not as diagnostic for us. These particular reactions occur in organisms that we typically don't use as unknowns, or that typically don't really have a lot of medical importance, for example. And so those are kind of the bell marks we're going to look for is, can your organism reduce nitrate to nitrite, or can it reduce nitrate to ammonia? And basically this just involves, we look for one other thing. Some organisms can directly reduce nitrate into nitrogen gas. So basically we will inoculate organisms into tubes of broth. Those tubes of broth will have a Durham tube in them. What's a Durham tube in the checking for? The nitrogen gas. Okay, so nitrogen gas accumulates in the tube, then we can say, yeah, that reduces nitrate to nitrogen gas. We're done. Okay. We have to give them nitrate in the broth because that's not a natural part of the broth mixture. And if we don't get any gas, then we've got a chemical test that determines whether nitrite has been produced. And then there is a chemical inference, we'll talk about that later on, that will allow us to determine whether ammonium has been produced. So this is going to be an important test later on in terms of identification of your unknown. Okay? Now, if your organism isn't facultative, then will it be able to do this? No. So the strict aerobe should be giving us a negative nitrate test. They just won't use that nitrate. They can't. Okay? They don't have that capability. Well, I'm not going to go through all these. These are really kind of for your purposes in terms of wrapping your head around the nitrogen cycle. But the next 2 or 3 diagrams show you kind of the complexity of the nitrogen cycle and how nitrogen is converted from one form into another as we go from the nitrogen gas in the atmosphere through the biosphere, through living organisms, including plants, animals, and microorganisms. And ultimately, that nitrogen is converted back into nitrogen gas again. And so there are elements of nitrogen fixation, which involve bacteria. There are processes of ammonification, which involves decomposition. And then we have uptake of this ammonia by plant roots to support their growth. But ultimately, that ammonia has got to be converted back into nitrogen gas again to keep the cycle going. So lithotrophs can convert it into nitrates. Facultative anaerobes can convert the nitrates into nitrites, and ultimately back into nitrogen gas again. So this self-sustaining cycle, which all life on planet Earth depends upon, is the result of microbial metabolism and basically one type of organism producing a product that another organism then uses as an energy resource. And by sequentially using products from one another, we can complete this cycle and make it self-sustaining. So most of the other cycles that we have operating in our worldwide biosphere, the phosphorus cycle, the sulfur cycle, the carbon cycle, all have a similar underlying basis. They're the result of microbial metabolism. And so that gets back to the question on the first exam. Dr. Evil would be destroying all of the natural recycling systems for nutrients in the ecosystem. Life on Earth just could not exist without these particular organisms. So it's pretty crucial and pretty important. And these are just different representations of the nitrogen cycle. You can take a look at some of those. Okay. So far we have been talking only about chemotrophs. Those organisms that get their energy from the oxidation of either organic molecules, or in the case we've just been talking about, inorganic molecules. Phototrophs. These are using sunlight as their source of energy. But remember, to characterize these organisms, we also have to specify what is their source of carbon. Their source of carbon is CO2. Then they are photoautotrophs. If it is some organic molecule that's being used as their source of carbon, then they're photoheterotrophs. We're not really going to deal with these at all. And we're going to spend just a couple of minutes talking about photoautotrophy. And we've already done a little bit of this as we've done the survey of the microbial world when we looked at Berkey's manual. Remember, in photoautotrophs, there are two types of photosynthesis. There's oxygenic photosynthesis, and there is anoxygenic photosynthesis. Which group does the oxygenic? Volume 1, Berkey's manual. Phylum Cyanobacteria. These are the cyanobacteria, the blue-green bacteria. And they really have the same type of photosynthetic system that a higher plant does. Okay? Two photosystems, photosystem 2, photosystem 1. Remember what happens in photosystem 2? Water is split. Those electrons are picked up by some type of an electron acceptor. And as the electrons are passed to photosystem 2, through an electron transport chain, we make our hydrogen ion gradient to make our ATP. And then in photosystem 1, those electrons are excited again and picked up by an electron acceptor and ultimately transferred to NADP to make reduced NADP. And then we use that ATP in the reduced NADP along with CO2 in the Calvin cycle to make carbohydrate. Vague memories? Distant memories? The main difference between these particular photosystems and those of higher plants is a slight variation in the pigments that make up these photosystems. And in particular, we have that cyanin pigment, which is one of the parts of the photosystems that absorbs just a slightly different wavelength of visible light than typically chlorophyll A or chlorophyll B does. So there's really minimal differences in those particular processes of oxygenic photosynthesis in bacteria and in higher plants. Where it gets a little bit interesting is when we get down here into this anoxygenic photosynthesis. And we're going to look at a couple of examples here. And look at the green and purple sulfur bacteria. And then we'll also look a little bit at the green and purple non-sulfur bacteria. Let's actually get these out of the way first. Green and purple non-sulfur bacteria are pretty bizarre because they are the photoheterotrophic organisms. So think about the implications of that for a second. If you're a photoheterotroph, you're using sunlight as your source of energy to make ATP, but you're using organic molecules as your source of carbon. So do you need a Calvin cycle if you do that? No, your carbon has already been fixed into organic forms for you. You're ingesting carbon in the form of an organic form. That's a pretty interesting strategy, to use that light energy for ATP production, but not using light energy to power carbon fixation. For the autotrophs, we are going to need carbon fixation occurring. One of the big differences between this anoxygenic photosynthesis that these guys do and the oxygenic photosynthesis that cyanobacteria does is that an anoxygenic photosynthesis, there aren't two photosystems. There's only a single photosystem. In some organisms, it's photosystem 1, and in some organisms, it's photosystem 2. We'll show you the difference in just a second. The other major difference is that the photosynthetic pigment is quite a bit different than typical chlorophyll. It's called bacteria chlorophyll. And one of the ways you can visually see the differences in these pigments is by looking at the absorption spectrum of chlorophylls and bacteria chlorophylls. And what's really kind of striking is these peaks of absorbance over here in the near infrared and even the infrared part of the spectrum. And you really don't get any absorption there by chlorophylls. Now you get some of the same absorption down here in the purple and blue wavelengths, but this is trapping some wavelengths of light that normally aren't available for other plants. One of the things I said was that microorganisms have been around for a long time. Earliest fossils of microorganisms we can trace back to the planet are about 3.5 billion years. In 3.5 billion years, a lot of evolution and adaptation can take place. And basically, as a group, prokaryotes have pretty much tried every strategy possible to fill a niche. And one of the ways that this is illustrated is in this photoautotrophy and anoxygenic photosynthesis. Some organisms have quote unquote kind of tinkered around with getting by with one photosystem. And sometimes it's photosystem 2, sometimes it's photosystem number 1. Cyanobacteria appear to have been more recently evolved, and they kind of took the best of both worlds and used the two photosystems together cooperatively. Okay? So it's really interesting to look at microorganisms to kind of see the different strategies they quote unquote tried to survive. Okay, so here's anoxygenic photosynthesis. In the purple sulfur bacteria. They only have one photosystem. It happens to be photosystem number 1. The light that's absorbed by this bacteria chlorophyll in photosystem number 1 is of a longer wavelength than we typically see absorbed in the photosystems of higher plants. Does longer wavelength have higher or lower energy? Longer the wavelength, the lower the energy. Okay. So basically, this kind of near infrared radiation doesn't have enough energy to split and break a water molecule bond. But it does have sufficient energy to split the bonds of other molecules like hydrogen sulfide, or maybe even hydrogen. Sometimes some organic compounds can even be split to generate electrons. And then what happens to those electrons? A proton gradient is formed, and the electrons are used to reduce NADP. So this NADP and the ATP can then be used in carbon fixation. But we constantly need a source of electrons coming in the form of some kind of reduced compound like hydrogen sulfide. Contrast that with the purple and green non-sulfur bacteria. Okay? These guys now are the photoheterotrophs. They only have a single photosystem, which is photosystem 2, and the type of electron flow that takes here is called a cyclic electron flow. And so the electrons basically are going from the photosystem to an electron transport chain and back to that same photosystem again. So the electrons basically are boosted, transferred to other compounds, and then go back to the same photosystem again. So it's a really different strategy involving a different photosystem, but again, the outcome is the same. They're able to sustain themselves using sunlight energy. So different strategies, different microorganisms. This is a good summary slide that kind of summarizes some of the things that we've been talking about, particularly in regard to characterizing microorganisms into those categories of photoautotrope, photoheterotroph, chemoautotrope, chemoheterotroph. So this is one you might want to take a look at to kind of summarize some of this material. So we're going to see a lot of follow-up from this information into lab, particularly after the first lab exam. We're going to be doing a lot of fermentation tests. We're going to be seeing if organisms can metabolize certain foods and what byproducts they'll produce as a result of the metabolism. So we're going to be coming back and kind of applying some of this stuff later on. Okay, I know many of you have a medical interest. That's why a lot of you are here. so far, the things we've been talking about, kind of the history of microbiology, classification of microorganisms, growth of microorganisms, have had to do with more taxonomy, with some more ecological considerations. And now we're going to start delving into more medically related things. Okay? So kind of where we're going to be going is the following. First thing we're going to look at is we've talked about factors that influence the growth of microorganisms and how these microorganisms have this incredible potential to grow and divide very rapidly. And that can have health consequences, obviously. So one of the first things we're going to do is talk about, okay, how do you go about controlling the growth of microorganisms? How do we remove them or eliminate them or inhibit them? Because from a medical standpoint, that can be important. You do that every day when you come into lab. Okay, you spray the Clorox spray on your tabletops right there to try to disinfect your tabletops so you won't get contamination. Well, why is that spray working? What's happening when you spray that on the countertop? Why is that particular substance antibacterial? We'll talk about the use of both physical agents like heat and filtration and radiation to control growth. We'll talk about a number of chemical agents that can be used. Some of those we mentioned very briefly in the history of microbiology. I talked about the fact that Lister used this stuff called carbolic acid or phenol to sterilize his surgical instruments. How did that work? Semmelweis used the chloride of lime for hand washing. How did that work? So we'll spend a fair amount of time talking about some of the chemical agents that are used routinely in eliminating microorganisms. Then we'll shift gears a little bit and talk about some of the things you can take internally, some of the drugs you can take. So we'll talk about antibiotics and other types of antibacterial agents which are designed for internal use versus external use. Then we're going to spend some time talking about the relationship between parasites and hosts, and specifically bacterial pathogens and the human body. And we're going to kind of look at, okay, what are the offensive capabilities that microorganisms possess that allow them to colonize your body and to multiply and to spread, and unfortunately, usually as a result of that, cause disease. So we're going to kind of look at the offensive capabilities of microorganisms and their disease-causing properties. And then finally, we'll wrap up with talking about the defense. What are your defensive mechanisms you have? What does your immune system look like? What are all those different kind of levels of immunity? And that gets pretty complicated and gets pretty hairy. And that's one of the reasons why in this class I don't give a comprehensive final. Your final exam is just a fourth exam because it's over immunology. And dealing with immunology is more than enough to handle for an exam, let alone having to go back and remember the stuff you did in the first three exams. So we'll keep you plenty busy with immunology. The very end part of the course, we'll talk about some applications to immunology, and we'll talk about some immunological testing, antibody-based tests, monoclonal antibodies, some serological tests, and then we'll be done. Okay, so we're going to really make this transition now to more kind of medically related information. Let me skip forward a slide or two. Ever since von Leeuwenhoek developed the first microscope, and people started looking at water and other things and found out that there were microorganisms living in them, people have understood that this microbial world existed. And a lot of people also thought and kind of intuited that maybe these microorganisms were somehow responsible for disease. But we know that wasn't firmly established until Koch and Koch's postulates in the 1860s. But I mean, people intuitively know that disease was transmissible, that certain infections were contagious just by observation. Okay? In the 13th century, when 2/3 of the people in Europe got the Black Plague and about half of them died, it didn't take a rocket scientist to look around and say, man, if there's sickness in the town, then I might get sick as well. And this particular picture represents kind of the horror that people expressed when they started looking at the Thames River in London, for example. Because what were they doing? Everybody was just discharging their sewage into the Thames River, and it was just becoming this breeding ground for bacteria and other prokaryotic organisms, many of which were pathogens. It's kind of interesting to look at the history of infectious disease and to see what methods were developed to try to prevent contamination and prevent infection from these agents of disease, even though we weren't absolutely sure what those agents were. For a long time, people thought that sickness was transmitted primarily in the form of what was called a miasma. And miasma basically means bad or foul air. And so people felt like, wow, if there's a sick person, they can release these miasmatic agents into the air by breathing. And if I breathe them, then I could get sick as well. But they didn't know for sure that they were bacteria or that they were protozoa. They thought in many cases it was kind of evil spirits. Okay? So there was a lot of kind of mythology surrounding prevention of infection. But what's interesting is some of the things that were developed and some of the techniques that were developed, even though the people had no idea what the cause of the disease were, actually turned out having some scientific basis. So for example, if you look at this plague clothing from 13th century Europe, there's some sound, fundamental antimicrobial strategy going on here, okay? The gown kind of helps prevent you from getting stuff on your skin. The scarf kind of covers up your mouth. And then this long beak-like protuberance right here might have been stuffed with cotton, for example, and had some air filtering capabilities associated with it. So, you know, who knows how people stumbled on these particular kind of strategies or garbs. But what they found out is sometimes they worked. And if they worked and you had a less chance of getting sick, other people noticed and they became kind of adopted. It wasn't uncommon for people to burn various forms of aromatic woods and incense to try to prevent infection. And again, the idea was that these aromatic woods and incense might destroy these miasmas that you could be breathing in. Well, as it turns out now, we know that sometimes when these aromatic woods and incense are born, they produce formaldehyde. And formaldehyde is a very potent antimicrobial agent. Were you familiar with formaldehyde? Preservation, embalming, okay. We used to do fetal pigs in formaldehyde. Very irritating. And unfortunately, we then found out that formaldehyde was a carcinogen. So that had to be replaced with other safer preservatives, which don't smell great but aren't nearly as irritating and as bad as formaldehyde is. In ancient civilizations, particularly in Rome and Greece, rich people used a lot of copper and silver dining and especially drinking utensils. The poor people had to use wooden vessels, for example, for drinking cups. What was so great about silver, gold, copper? Well, they were pretty and they kind of showed status and things like that. But again, people noticed that drinking out of these particular vessels meant less chance of waterborne contamination than drinking out of a wooden vessel. Why? Because those heavy metals, that copper, the iron, the gold, have antibacterial properties. And so sometimes just by blind luck or happenstance, people stumbled onto things that were effective long before they knew what the causes of the particular disease were. One of the things that is going to be important to get under your belt early on is some terminology. So we're all sure that we're speaking the same language. Because I'm going to make routine references to disinfection and antisepsis and sterilization and sanitization and pasteurization. Okay? So this is one thing I would like you to look at for Thursday is look through this particular slide and familiarize yourself with what these definitions are, and then we will be using these as we talk about examples of various types of chemical agents used to control microbial growth. This particular slide right here too is a good one, and it's one that I would probably kind of keep handy, because this gives us a nice flow diagram to kind of break down and categorize microbial, antimicrobial agents. So you'll see, for example, there's 3 boxes here. There are physical agents, there are chemical agents, there are mechanical agents right here. And then that further breaks them down into heat, radiation. Heat is broken down into dry and moist. Moist heat is broken down into pressurized moist heat versus non-pressurized moist heat. So this is really a good way to kind of organize and kind of deal with the complexity of some of the stuff we're going to talk about. So that's where we're going to pick up Wednesday. So remember, rock unknown agriculture. We're going to be working in groups of 3 or 4 tomorrow to inoculate known organisms. So I'll tell you kind of what your group is going to be tomorrow.